While working in the molecular biology laboratory, we extract different kinds of biological molecules. In order to analyze the expression of genes implicated in different disorders or the detection of RNA viruses like coronavirus or any other downstream technology, we first have to extract the RNA. There are different methods to extract the RNA, like the kit method, triazole method, or the manual method. But the triazole method is the most commonly used method while performing the extraction of RNA in molecular biology laboratory. So in this practical, we will demonstrate that how to extract the RNA from blood using triazole method. Let's perform the experiment.
The major reagents and materials required for RNA extraction are blood, triazole reagent. Triazole reagent is a light sensitive chemical and it preserves the integrity of RNA. Next is chloroform, isopropanol, ethanol, and finally nuclease-free water. First of all, 250 microliter blood sample will be taken in Eppendorf tube. After the addition of blood sample, 750 microliter of triazole reagent will be added. Once you will add the triazole reagent, Eppendorf tube will be inverted to mix the samples homogeneously. Next, 200 microliter of chloroform will be taken and add in the Eppendorf tube. The function of chloroform is to separate the RNA from other cell contents and proteins. After the addition of chloroform, vortex the opened off tube for 1 minute. In next step, we will centrifuge the sample at 13,000 rpm for 15 minutes at 4 degrees Celsius. After the completion of centrifugation, you will take out the opened off tube . As a result of centrifugation, you will observe 3 layers. Upper is the aqueous phase that contain the RNA. Second is the interphase, and third are the impurities. Now we will transfer the aqueous phase to a new opened off tube . It should be noted that the tip of the pipette should not touch the interphase. You will just pick the aqueous phase that contain the desired RNA molecule. After the transfer of aqueous phase to new opened off tube, we will add the equal volume of isopropanol in the opened off tube to precipitate the RNA. Similarly, after the addition of isopropanol, we will again centrifuge the sample at 13,000 rpm for 15 minutes. After centrifugation, you will observe the pellet in the bottom of the tube and you will discard the supernatant, preventing the pellet. Now we will wash the RNA pellet with ethanol to remove the impurities. So 500 to 600 microliter of 70% chilled ethanol will be added into the Eppendorf tube. After the addition of ethanol, we will again centrifuge the samples at 13,000 rpm for 15 minutes. After the centrifugation, discard the supernatant. Finally, we will add nuclease-free water into the Eppendorf tube after air drying the ethanol. The nuclease-free water will protect the RNA.
Finally, after the addition of nuclease-free water, we will store the RNA at -80 degrees Celsius.